Hello students, welcome to my channel Online Biology Tutorials. In today's live, I will be reading Chapter 5 Molecular Basis of Inheritance. 4 letters, infinite meaning. Today, we will read the language written inside every cell. Imagine a spiral staircase glowing in the dark. 3, 2, 1. Let's begin. This chapter belongs to the second unit of class 12th NCERT Biology, one of the mega chapter of this book. Very important. Let's begin. In the previous chapter, you have learned the inheritance patterns and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material, at least for the majority of organisms. In class 11, you have learned that nucleic acids are polymers of nucleotides. Deoxyribonucleic acid or DNA and ribonucleic acid RNA are the two types of nucleic acids found in living systems. DNA acts as the genetic material in most of the organisms. RNA, though, it also acts as a genetic material in some viruses. Mostly functions as a messenger, RNA has additional roles as well. It functions as adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned the structures of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, that is transcription, the genetic code that determines the sequences of amino acids in proteins, the process of protein synthesis, translation, and elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent sections, we will understand that why it is the most abundant genetic material and what is the relationship is with RNA. So we begin with 5.1, the DNA. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotides referred to as base pairs present in it. This also is the characteristic of an organism. For example, a bacteriophage known as 5X174 has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs or BP. Escherichia coli has 4.6 into 10 to the power 6 base pairs, and the haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Let us discuss the structure of such a long polymer. (5.1.1) Structure of polynucleotide chain. Let us recapitulate the chemical structure of polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group. There are two types of nitrogenous bases: purines, example, adenine and guanine, and pyrimidines, example, cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the time of thymine. A nitrogenous base is linked to the OH of carbon 1 pentose sugar through the N-glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine. When a phosphate group is linked to OH of carbon 5 of a nucleoside through phosphoester linkage, a corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present, is formed. Two nucleotides are linked through 3 to 5 prime phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at 5 prime end of sugar, which is referred to as 5 prime end of a polynucleotide chain. Similarly, at the other end of the polymer, the sugar has free OH of 3 prime carbon group, which is referred to as 3 prime end of the polynucleotide chain. The backbone of a polynucleotide chain is formed due to sugar and phosphates. The nitrogenous bases linked to sugar moiety project from the backbone. So it's the time to write the keywords. Write any 5 keywords that are remembered by you from the text read so far. Go ahead. 1, 2, 3. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. Also in RNA, the uracil is found at a place of thymine, 5-methyl uracil, another chemical name for thymine. DNA as an acidic substance present in nucleus was first identified by Frederick Miescher in 1869. He named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, produced a very simple but famous double helix model of the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and always equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. Also, if each strand from a DNA, let us call it as a parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as daughter DNA, thus produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA became very clear. So, whatever keywords you have retained till now, type any two in the chat and proceed further. The salient features of the double helix structure of DNA are as follows. It is made of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. The two chains have anti-parallel polarity. It means if one chain has the polarity of 5 to 3 prime, then the other has 3 to 5 prime. The bases in two strands are paired through hydrogen bond, forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.5 nanometer. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meter, and there are roughly 10 base pairs in each turn. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. Compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? The proposition of a double helix structure of DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA to protein. DNA is self-replicated. From DNA, RNA is formed, which is known as transcription. From RNA, proteins are being formed, which is known as translation. This is known as central dogma of biology. May I know in the chat, what is the site of transcription? Quickly write and we will move further. In some viruses, the flow of information is in reverse direction, that is, from RNA to DNA. Can you suggest a simple name to the process? 5.1.2 Packaging of DNA helix. Take the distance between two consecutive base pairs as 0.34 nanometer. If the length of DNA double helix is a typical mammalian cell is calculated, so simply by multiplying the total number of base pair with the distance between two consecutive base pair, that is 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9 base pairs, meter per base pair, it comes out to be approximately 2.2 meters, a length that is far greater than the dimension of a typical nucleus, approximately 10 to the power minus 6 meter. How is such a long polymer packed in a cell? If the length of E. coli DNA is 1.36 millimeter, can you calculate the number of base pairs in E. coli? In case you want to know the answer to this question, you can come to the playlist of my channel with the name Molecular Basis of Biology, and there you will find a video that will give you step-by-step calculation of this numerical problem. In prokaryotes such as E. coli, though they do not have a definite nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positively charges in a region termed as nucleoid. The DNA is nucleoid is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acids residues with charged side chains. Histones are rich in the basic amino acid residues lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pairs of DNA helix. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. Thread-like stained colored bodies seen in the nucleus, the nucleosomes in chromatin, are seen as beads on string structure when viewed under electron microscope or abbreviated as EM. Theoretically, how many such beads or nucleosomes do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packed to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form the chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal proteins. In a typical nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are known as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Type any 3 terms that you have learned so far from the paragraph that has just been read. Five point two. The search for genetic material. Even though the discovery of nuclein by Miescher and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumonia, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism or bacteria had changed in physical form. When Streptococcus pneumonia, pneumococcus bacteria are grown on a culture plate, some produce smooth, shiny colonies, while others produce rough colonies. This is because the S strain bacteria have a mucus or polysaccharide coat, while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. S strain, when injected into the mice, mice died, whereas when R strain was injected into the mice lived. Griffith was able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. So, his observations were when heat killed S strain was injected into the mice lived. But when heat killed S strain was mixed with the live R strain injected into the mice died. When he injected a mixture of heat killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of the genetic material was not defined from his experiments. Biochemical characterization of transforming principle. Prior to the work of Oswald Avery, Colin MacLeod, and Macklin McCarty, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals like proteins, DNA, RNA from the heat-killed S strains to see which ones could transform live R strains into the S strains. They discovered that DNA alone from the S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes called proteases and RNA digesting enzymes RNases did not affect transformation. So the transforming substance was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is a hereditary material, but not all biologists were convinced. Can you think of any difference between DNA and DNAs? Yes, it's a time to write keyword. Quickly write any two keywords retained by you by the reading that has been done so far. Go ahead. Five point 2.1. The genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which were infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria. 5.2.2 Properties of genetic material, DNA versus RNA. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from the Hershey-Chase experiment. It became an established fact that it is DNA that acts as a genetic material. Became clear that in some viruses, RNA is the genetic material, for example, tobacco mosaic viruses, QB bacteriophage, etc. Answer to some of the questions such as why DNA is the predominant genetic material whereas RNA performs dynamic functions of messenger and adapter has to be found from the differences between the chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule that can act as a genetic material must fulfill the following criteria. It should be able to generate its replica or replication should be done by it. It should be stable chemically and structurally. It should provide the scope for slow changes or mutation that are required for evolution. It should be able to express itself in the form of Mendelian characters. If one examines each requirement one by one because of the rule of base pairing and complementarity, both the nucleic acids DNA and RNA have the ability to direct their duplications. The other molecules in the living system such as proteins fail to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle. Itself that heat which killed the bacteria at least did not destroy some of the properties of the genetic material. This now can easily be explained in the light of the DNA that the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, 2' OH group present at every nucleotide in RNA is a very reactive group and makes RNA labile and easily degradable. RNA is also known as catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins and hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material. But DNA being more stable is preferred for storage of genetic material. For the transmission of genetic information, RNA is better. So, whatever keywords you have imbibed till now, do write any two keywords from the genetic material reading. Hi, Professor F.A. Hello, ma'am. My name is Ansh from Unacademy NEET UG dropper students. What is amazing teaching? What an amazing teaching style, ma'am. Thank you for the amazing lecture. Thank you, dear. This is just the reading. In case you want to have elaborative videos, you can come to the playlist section of this chapter on my channel. There we have several videos on the topics that are being given in NCERT. 5.3 RNA World. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? It should be discussed in detail in the chapter on chemical evolution. But briefly, we shall highlight some of the facts and points. RNA was the first genetic material. This is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that made it more stable. DNA being double-stranded and having complementary strand further resist changes by evolving a process of repair. Can I request you, Ansh, to please share this video with your friends? In case anybody wants to have revision just prior to 27th of March, they can go through these videos. They will definitely help in retaining each word. 5.4 Replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication that has been shown to you in figure 5.6. Next topic, 5.4.1, the experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms such as plants, human cells. Matthew Michelson and Franklin Stahl performed the following experiment in 1958. They grew E. coli in a medium containing ammonium chloride. N15 is the heavy isotope of nitrogen. As the only nitrogen source for many generations, the result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. Please note that nitrogen 15 is not a radioactive isotope and it can be separated from N14 only based on densities. Then they transferred the cell into the medium with normal N14 ammonium chloride and took samples at various definite time intervals as the cell multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently as cesium chloride gradients to measure the densities of DNA. Can you recall what centrifugal force is and think why a molecule with higher mass or density would sediment faster? Thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, second generation, was composed of equal amounts of its hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid densities DNA molecule? Very similar experiments involving use of radioactive thymine to detect distribution of newly synthesized DNA in the chromosomes was performed on Vicia faba beans by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicates semi-conservatively. Time to write keywords. Write any 3 keywords imbibed so far. Go ahead. Five point 4.2 The machinery and the enzymes. In living cells such as E. coli, the process of replication requires a set of catalysts or enzymes. The main enzyme is referred to as DNA dependent DNA polymerase. Since it uses DNA template to catalyze the polymerization of deoxynucleotides, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 6 base pair, compare it with human whose diploid content is 6.6 into 10 to the power 9 base pair. Completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleoside triphosphates serve dual purposes. In addition to acting as substrate, they provide energy for polymerization reaction. The 2 terminal phosphates in a deoxynucleotide triphosphates are high energy phosphates, same as in case of ATP. In addition to DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the 2 strands of DNA cannot be separated in its entire length, the replication occurs within a small opening of the DNA helix referred to as replication fork. The DNA-dependent DNA polymerase catalyzes polymerization only in one direction, that is 5 to 3 prime. This creates some additional complications at the replication fork. Consequently, on one strand, the replication is continuous, while on other strand, the template with polarity 5 to 3 prime, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at the S phase of the cell cycle. The replication of DNA and cells division cycles should be highly coordinated. A failure in cell division after DNA replication results into polyploidy, a chromosomal anomaly. You will learn the detailed nature of origin, the process occurring at this site in higher classes. 5.5 Transcription. The process of copying genetic information excuse me, from one strand of the DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Why both the strands are not copied during transcription has a simple answer. First, if both strands acts as a template, they would code for RNA molecule which is different sequences. And in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complement the genetic material transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other and hence would form a double-stranded RNA. This would prevent RNA from being translated into protein and the exercise of transcription would become a futile one. 5.5.1 Transcription Unit. A transcription unit in DNA is defined primarily by the three regions in the DNA: a promoter, the structural gene, a terminator. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5 to 3 prime. The strand that has polarity 3 to 5 prime act as a template and it is also referred to as the template strand. The other strand which has the polarity of 5 to 3 prime and the sequence same as RNA except thymine at the place of uracil is displaced during transcription. Strangely, this strand is referred to as the coding strand. All the reference point while defining a transcription unit is made with coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. You need to read this sequence from the NCERT book and you should practice as well. For example, if a coding strand sequence is being given or a template strand is given to you, you have to deduce either of the one from the given sequence. So you should know how to do it. The promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5' end upstream of the structural gene. The reference is made with respect to the polarity of coding strand. It is the DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strand. By switching its position with terminator, the definition of coding and template strands could be reversed. The terminator is located towards 3' or downstream of the coding strand and it usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. 5.5.2 Transcription Unit and the Gene. A gene is defined as the functional unit of inheritance, though there is no ambiguity that the genes are located on the DNA. It is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for the transfer RNA or ribosomal RNA molecule also defines a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said monocistronic, as in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or express sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. 5.5.3 Types of RNA and the process of transcription. In bacteria, there are three major types of RNAs: messenger RNA, transfer RNA, and ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The messenger RNA provides the template, tRNA brings amino acids and reads the genetic code, and ribosomal RNA plays structural and catalytic role during the translation. There is single DNA dependent RNA polymerase that catalyzes transcription of all the types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription or initiation. It uses nucleoside triphosphates as substrates and polymerizes in a template dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminated region, the nascent RNA falls off. So also the RNA polymerase. This results in termination of transcription. An intriguing question is that how is the RNA polymerase is able to catalyze all the three steps, which is initiation, elongation, and termination. The RNA polymerase is also capable of catalyzing the process of elongation. It associates transiently with the initiation factor or the sigma factor and termination factor, which is the rho factor, to initiate the termination of the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the messenger RNA does not require any processing to become active, and also since transcription and translation take place in the same compartment, many times the translation can begin much before the messenger RNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. There are at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. The RNA polymerase 1 transcribes ribosomal RNA, whereas RNA polymerase 3 is responsible for the transcription of transfer RNA, 5 small ribosomal RNA and small nuclear RNA, the RNA polymerase II transcribes precursor of messenger RNA, the heterogeneous nuclear RNA. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Hence, it is subjected to a process called splicing, where the introns are removed and exons are joined in a definite order. Heterogeneous RNA undergo additional processing called as capping and tailing. In capping, an unusual nucleotide, which is methyl guanosine triphosphate, is added to the 5' end of heterogeneous RNA. In tailing, adenylate residues 200 to 300 in number are added at 3' end in a template independent manner. It is the fully processed heterogeneous RNA, now called messenger RNA, that is transported out of the nucleus for translation. The significance of such complexities is now beginning to be understood. The split gene arrangements represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA and RNA-dependent processes in the living system have assumed more importance. 5.6 Genetic Code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor it could any be drawn theoretically. There existed ample evidences, though, to support the notion that change in nucleic acid were responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition of deciphering of genetic code were most challenging. In a very true sense, it requires the involvement of scientists from several disciplines: physicists, organic chemists, biochemists, and geneticists. It was George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made of 3 nucleotides. This was very bold proposition because a permutation combination of 4 to the power of 3 would generate 64 codons, generally many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases, homopolymers and copolymers. Marshall-Nirenberg cell-free system for protein synthesis finally helped the code to be deciphered. Severo-Ochoa enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template-independent manner. Finally, a checkerboard for genetic code was prepared, and it is given in NCERT. Do prepare how to prepare this complete table with 64 codons, out of which 61 are the coding ones and 3 are the terminating ones. Question may come on it. Do prepare for it. It will help you in NEET as well. The salient features of genetic code are as follows. The codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acid. Hence, they function as stop codons. Some amino acids are coded by more than one codon, hence the code is degenerate. The codon is read in messenger RNA in a contiguous fashion. There are no punctuations. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. AUG has dual functions. It codes for methionine, which is the initiator codon. UAA, UAG, UGA are stop terminator codons. If following is the sequence of nucleotides in messenger RNA, predict the sequence of amino acid coded by it, take help of the checkerboard. So here AUG is given. If you see in the table, AUG is coding for methionine. If you see for UUU, UUU is just written above, it is phenylalanine. UUC, it is again phenylalanine, UUC phenylalanine, UUU phenylalanine, UUU phenylalanine, UUC is phenylalanine. Make sure that if you are writing amino acids, you need to use these three letters that are given in the genetic code table. Now try the opposite. Following is the sequence of amino acids coded by an messenger RNA. Predict the nucleotide sequence in the RNA. So the amino acids that are being given are methionine, phenylalanine 6 times. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learned? 5.6.1 Mutations and genetic code. The relationships between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 4. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so as function. The effect of point mutations will be explained here. A classical example of point mutation is a change of single base pair in the gene of beta globin chain that results in the change of amino acid residue glutamate to valine. It results into a diseased condition called sickle cell anemia. Effect of point mutation that inserts or deletes a base in structural gene can be better understood by following simple example. So this example requires visualization. Better to read all the statements that they have given in the book. And in case you find any difficulty, you can come to the playlist of this chapter on my channel. There is a video that has explained you in detail the different types of mutations, namely missense, nonsense, frame shift mutations, and silent mutations as well. The conclusion from the above exercise is very obvious. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutations. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards. 5.6.2 Transfer RNA, the adapter molecule. From the very beginning of the proposition of code, it was clear that Francis Crick that there has to be a mechanism to read the code and also to think and link all the amino acids. Because amino acids have no structure specialities to read the code uniquely, he postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind to specific amino acids. The transfer RNA, then also called soluble RNA, was known before the genetic code was postulated. However, its code as an adapter molecule was assigned much later. tRNA has an anti-codon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acid. tRNAs are specific for each amino acid. For initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNAs for stop codons. So, in the figure that has been given in the NCERT figure 5.12, the secondary structure of tRNA has been depicted that looks like a clover leaf. In actual structure, the tRNA is a compact molecule which looks like the inverted L. 5.7 Translation. Translation refers to the process of polymerization of amino acids to form polypeptide. The order and sequence of amino acids are defined by the process of bases in the messenger RNA. The amino acids are joined by a bond which is known as peptide bond. Formation of a peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as the charging of tRNA or amino acylation of tRNA to be more specific. If two such charged RNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins in its active state. It exists as two subunits, a large subunit and a small subunit. When the small subunit encounters a messenger RNA, the process of translation of the messenger RNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst. 23S ribosomal RNA in bacteria is the enzyme ribozyme for the formation of the peptide bond. A translational unit in messenger RNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. And messenger RNA also has some additional sequences that are not translated and are referred to as untranslated regions or UTR. The UTR are present at both 5' end before start codon and at 3' end after the stop codon. They are required for efficient translation process. For initiation, the ribosome binds to the messenger RNA at the start codon AUG that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in messenger RNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the messenger RNA. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by messenger RNA. At the end, a release factor binds to the stop codon, terminating the translation and releasing the complete polypeptide from the ribosome. So we are done with the last step of the central dogma of biology. Whatever three important terms you are able to retain from the translation, do mention it in the chat and then move further. I am moving to the next topic, that is 5.8 from your NCERT, regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcriptional level, processing level, transport of messenger RNA from nucleus to cytoplasm, or at translational level. The genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, If the bacteria do not have lactose around them to be utilized for energy source, there would be no longer required the synthesis of the enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic, physiological, or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also a result of coordinated regulation of expression of several sets of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize the start sites. These regulatory proteins can act both positively and negatively. The accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. The operator region is adjacent to the promoter elements in most operons, and in most cases the sequences of operator bind a repressor protein. Each operon has its specific operator and specific repressor. For example, LAC operator is present only in the LAC operon and it interacts specifically with LAC repressor only. 5.8.1 The LAC operon. The elucidation of the LAC operon was also a result of a close association between a geneticist Francois Jacob and a biochemist Jacques Monod. They were the first to elucidate a transcriptionally regulated system. In LAC operon, here LAC refers to lactose, a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as operon. To name very few such examples: LAC operon, tryptophan operon, arabinose operon, histidine operon, valine operon, etc. The LAC operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor. The three structural genes Z, Y and A. The I gene codes for the repressor of the LAC operon. The Z gene codes for beta galactosidase or BGAL, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units galactose and glucose. The Y gene codes for the permease, which increases the permeability of the cell to beta galactosidase or beta galactosides. The A gene codes for transacetylase. Hence, all the three genes products the LAC operon are required for metabolism of the lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. The illustration that has been given for the LAC operon is no doubt very important. In case any question comes on the LAC operon, this is must to be drawn. Lactose is the substrate for the enzyme beta galactosidase and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of inducers such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of LAC operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot act as inducers for lac-operon. Can you think of how long the lac-operon would be expressed in the presence of lactose? Regulation of lac-operon by repressor is termed as negative regulation. Lac-operon is under control of positive regulation as well, but that is beyond the scope of discussion at this level. Next, 5.9, Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in the DNA that determines the genetic information of a given organism. In other words, genetic makeup of the organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project or HGP was known as the mega project because you can imagine the magnitude and the requirements for the project if we simply define the aims of the project as follows. Human genome is said to have approximately 3 into 10 to the power 9 base pairs, and if the cost of sequencing required is $3 per base pairs, the total estimate cost of the project would be approximately 9 billion US dollars. Further, if the obtained sequences were to be stored in typed form in books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from the single human cell. The enormous amount of DNA expected to be generated also necessitated the use of high speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of the new area in biology called bioinformatics. Goals of HGP. Some of the important goals of HGP were as follows: identify all the approximately 20,000 to 25,000 genes in the human DNA, determine the sequence of the 3 billion chemical base pairs that make up the human DNA, store this information in databases, improve tools for data analysis, transfer related technologies to other sectors such as industries, address to the ethical, legal, and social issues that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of HGP, the Wellcome Trust of UK became the major partner. Additional contributions came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effects of DNA variation among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect the human beings. Besides providing clues to understanding human biology, learning about non-human organisms' DNA sequences can lead to an understanding of their natural capabilities that can be applied toward solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Rhabditis elegans, a free-living non-pathogenic nematode, Drosophila the fruit fly, plants like rice, Arabidopsis have also been sequenced. Methodologies. The method involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as express sequence tags or ESTs. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence and later assigning different regions in the sequence with functions. a term referred to as sequence annotation. For sequencing, the total DNA from the cell is isolated and converted into random fragments of relatively smaller sizes and cloned in suitable hosts using specialized vectors. The cloning resulted into amplification of each piece of the DNA fragment so that it subsequently could be sequenced with ACE. The commonly used hosts were bacteria, yeast, and the vectors were called as BAC or bacterial artificial chromosomes and YAC yeast artificial chromosomes. The fragments were sequenced using automated DNA sequences that worked on the principle of a method developed by Frederick Sanger. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome number 1 was completed only in May 2006, means in 2006. This was the last of 24 chromosomes, 22 autosomes, and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease, recognition sites, and some repetitive DNA sequences known as microsatellites. So, the salient features of Human Genome Project. Some of the salient observations drawn from Human Genome Project are: human genome contains 3164.7 million base pairs. The average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. The total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 1,40,000 genes. Almost all nucleotide bases are exactly the same in all humans. The functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for proteins. Repeated sequences make up very large proportions of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has most genes, 2968, and Y has the fewest, 231. Scientists have identified about 1.4 million locations where single base DNA differences or SNPs or single nucleotide polymorphism occur in humans. This information promises to revolutionize the processes of finding chromosomal locations for disease associated sequences tracing human history. Applications and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decade, leading to our understanding of biological systems. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having the HD sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or few genes at a time. With whole genome sequences and a new high throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. So ultimately, I have reached to the final topic of this chapter 5.10, DNA fingerprinting. As stated in the preceding section, 99.9% of base sequence among humans is the same. Assuming human genome has 3 into 10 to the power 9 base pair, in how many base sequences would there be differences? Is it is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 6 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of any of the two individuals. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called the repetitive DNA because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks of during um, density gradient centrifugation. The bulk DNA forms a major peak, and the other small peaks are referred to as satellite DNA. Depending on base composition, length of segment, and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for any proteins, but they form a large proportion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicles, skin, bone, saliva, sperm from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism is variation of the genetic level arises due to mutation. New mutations may arise in an individual either in somatic cells or in the germ cells. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to other members of the population through sexual reproduction. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant or allele at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher as mutations in these sequences may not have any intermediate or immediate effect impact in individuals' reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of variability or polymorphism. There is a variety of different types of polymorphism ranging from single nucleotide change to a very large scale change. For evolution and speciation, such polymorphisms play a very important role, and you will study these details in higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used the satellite DNA as a prop that shows very high degree of polymorphism. It was called variable number of tandem repeats or VNTR. The technique as used earlier involved southern blot hybridization using radio labeled VNTR as a prop. It included isolation of DNA, digestion of DNA by restriction endonucleases, separation of DNA fragments by electrophoresis, transferring or blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. Hybridization using labeled VNTR probe and detection of hybridized DNA fragments by autoradiography. A symmetric representation of the DNA fingerprinting has been shown to you in figure 5.16. The VNTR belongs to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The number of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kilobase pair. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual. It differs from individual to individual in a population except in the case of monozygotic twins. The sensitivity of the technique has been increased by use of PCR or polymerase chain reaction. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider applications such as in determining population and genetic diversities. Currently, many different probes are used to generate the DNA fingerprints. So after reading of complete 1 hour and few minutes, the molecular basis of inheritance is completed, one of the major chapter of your book. In case you feel issue with any of the subtopic that is not understood by you, you are free to comment me in the chat and definitely I'll send you a link in solution to that. In case this video was helpful, do consider subscribing and share it with your friends so that they can also avail the benefit.